All right, let's get started with antimicrobial peptides. This is the part where we will be discussing the chemical mediators of immune system. So we'll start with this antimicrobial peptides, and let's start with the chemical nature of the antimicrobial peptides. Basically, these are cationic peptides. So they have that positive charge, and that's really important for their function. Important point is they are conserved peptides. The sequence remains highly conserved. Okay, and the action of these antimicrobial peptides is basically they are going to target the outer membrane of the bacteria right over there. So plasma membrane is the target of these antimicrobial peptides. They basically work in a different manner. So let me create the illustration of plasma membrane. Right, we know that in plasma membrane we have these phospholipids where we have fatty acids and those are present in the membrane. What they do is they perform electrostatic interaction with those molecules. Next is they form ionic pores, and because of those pores the molecules can come out. They are able to create membrane gaps, these peptides. Further, they are able to increase the membrane permeability. Because of that, the content from the bacterial cell will come out and eventually lysing the cell. So this is how these antimicrobial peptides, they work. Let's take the example, the first one, which is Cathelicidins. Okay, very important peptide, and this peptide is alpha helical in nature. So it's alpha helical peptide where you have, I'm sure you have seen the secondary structure of protein, right? Primary structure and secondary structure of the protein, you have helix, beta sheet. So this is the alpha helix. And what it don't have is cysteine amino acids. So that's a feature of this particular peptide. It doesn't have cysteine amino acid. Okay. And next is the total sequence of this particular peptide is 12 to 80 amino acids, depending upon different types of these cathelicidins. Right. So what are the cells that are going to produce this particular peptide? Let me draw the illustration. So this is cell number 1. This is cell number 3 that I'm designing. I don't know whether you can recognize these cells. The first one is neutrophils. Neutrophils are really important for our immune system. Next one is your respiratory epithelial cells. They're also able to create or produce these peptides. Alveolar macrophages in our lungs. We need alveolar macrophages that can produce these antimicrobial peptides, right? So this is highly important for our immune system, antimicrobial peptides. Second example of antimicrobial peptide, let's write it down. Defensins, as the name indicates, defensins, it can defend our system. These are open-ended peptides. That is the important feature of these peptides, open-ended, and then they're rich in amino acids like arginine and cysteine. Cysteine was absent in the first case, right? They form disulfide bonds. Because of that, its structure is really stable. You have molecular weight which is 4,000 Dalton is the molecular weight, and in that case you have beta sheets. Okay. And I'm trying to create the illustration of, let me draw it, beta sheets, right? So you have specifically anti-parallel beta sheets in this particular peptide. Just a little bit more complex than the first peptide. So let me write it down, anti-parallel beta sheets. Second one is, it also has beta-hairpin loop structures. So loop structures are also there. And again, it is cationic amino acids are there, right? In this particular antimicrobial peptide. If we talk about types, you have alpha defensins, and based on their molecular weight, I'm categorizing them. You have 29 to 35 amino acids in this case. Then second one is beta defensins. You have 36 to 42. These are larger antimicrobial peptides. So if we talk about the cells, again, our neutrophils and specifically granules inside the neutrophils, they contain this particular peptide. These are the primary granules that are green in color that are showing you the production of these peptides. Intestinal, in intestine, you have specific cells, Paneth cells. They can also produce this. Also, intestinal epithelial cells, they are also able to produce these peptides. These are the sources from where we get these particular peptides. And then respiratory epithelial cells are also able to produce these peptides, right? So in respiratory system, we need protection like using these particular peptides. So, and also intestine and also in our immune system like neutrophils. So they are really important. Now let's talk about histatins, right? another antimicrobial peptide. They are really large antimicrobial peptides, and they are composed by specific amino acids. And it can be there in human saliva. It has antifungal activity, and the amino acids that are present is around 24 to 28 amino acids. They are usually histidine amino acid rich molecules. Next is no ionic. They're not able to form the ionic channels. So that's, separate characteristic of these peptides. And it's going to target a specific organelle. Can you recognize that? That's the mitochondria. ATP producing organelle, mitochondria. They're going to target the mitochondria, and the mitochondria is essential for various structures, and that's why antifungal activity is there, right? And if you talk about other natural antimicrobial products, you have various components, like you have histone proteins, you have lactoferrin, second one lactoferrin, and you have also chemokines. They have the ability to actually kill the microbes. So these are some of the important components. Now let's move on to the next, where now we are going to discuss another component, very important component, which is bacteriocins. Right? What are bacteriocins? We can understand in detail their function. So let me write it down. Bacteriocins. Now, bacteriocins are basically the molecules that are produced by the beneficial bacteria, the normal microbiota, good bacteria. So this green in color, these are the bacteria that are able to produce these larger proteins, and these are not peptides. These are antimicrobial protein produced by normal microflora. So these are not peptides, these are antimicrobial proteins. And these proteins, they are highly toxic. They are highly toxic against pathogenic strains of the bacteria, right? So pathogenic strains, they are getting targeted with these antimicrobial peptides. They can target strains, they can target different species, but they are not going to affect the normal microbiota, interestingly. So if we talk about their function, biological barrier, they work as a biological barrier against invading microbes. They are going to protect our body. They are not going to allow the entry of these pathogenic strains, and they also control the competition, right? If bacteria is trying to outgrow the normal bacteria, it's going to target that. Characteristics: 900 to 5800 Dalton. That's the size. They are cationic in nature. They can be neutral also. They can also be anionic in nature. So different categories are there. Now let's talk about functions of bacteriocins. What are the different functions? One is they provide adaptive advantage to our body, because now the other bacteria is producing molecules that can give us adaptive advantage fighting against the infectious agent. They're gonna normally do not affect the good bacteria. Okay, so good bacteria are naturally immune against these particular proteins. And one of the interesting point is they may target, let me write it down, they may target the host cells also. There are some cases where these proteins, they're able to target the host cells. So specifically mononuclear phagocytes are getting targeted by these. So if diagrammatically, let me show this is the protein, and they can also target host cells. So that's no, they can actually kill these cells, some cells in some cases, but they are not going to kill the beneficial bacteria because bacteria is immune against those. So there will be no effect. If we talk about examples, now we need to also know the examples of bacteriocins. We have different bacteriocins, different functions. We'll take the example of, let's say, E. coli. In E. coli, we have colicins coming from coli. Colicins, different colicins are there. There are some genes that produce these. I'm listing all those genes. You have col B, you have col E1, you have col E2, then you have col 1, col 4. There are so many genes are there that are going to produce different types of bacteriocins, right? Now let's talk about action of colicins, how they actually work. Very interesting mechanism of action. So let me draw the bacteria first, and then I'll show you the targets. Different targets are there. This is the DNA in the center. Now these can target specific receptors. In bacteria, we have different receptors. So these can bind to these receptors. So bind to specific receptors that are present on the bacteria and affect their function. Next is they're gonna lyse the membrane. So as you can see over here, content can come out, they're going to target the membrane, and then lysis can happen. Next is they can target ribosomes. These green color structures that I'm drawing, these are ribosomes. And these proteins can target ribosomes, and the protein synthesis is going to get affected. That's another mechanism. They can also target the energy production. In bacteria, you have mesosomes. They can target energy production by targeting the mesosomes, right? If we talk about gram-positive bacteria, which are producing those kind of molecules, different molecules, like you have antibiotics that are being produced by streptococcus. Some of the examples are there that can give us protection. You have bacillus producing antibiotics. You have lactococcus producing these molecules. You have staphylococcus, like Staphylococcus epidermidis is one of the important organisms that can produce these protective molecules. So they are produced by beneficial microbes, that is, yes, and then basically, they are beneficial for the human host. So these are the features of these, specifically the Bacteriocins. So in this video, I try to conclude the important antimicrobial molecules, peptides, proteins. I hope you are able to understand all those things, and it gives you a starting point to actually dig deeper into this particular content. A lot of questions sometimes comes from this particular topic, and I want you to prepare for that in detail, right? It's really important as far as the exam is concerned. It's really important as far as competitive exam is concerned, right? So all right, so this is an important part of our innate immunity course, and I want you to also check out other courses on Research Goal. Please check them out. In this course, we are providing so much content. You're getting the notes, you're getting MCQs, and unfortunately they are not available on YouTube. YouTube, you only have the lecture content. For notes and MCQs, you have to go to Research Goal and then download all those things. And these are going to help you in your exam, prepare you better for those competitive as well as class exams, right? All right, so we'll meet in the next one, where we will be discussing other components of the immune system. All right.